The ARVO+Genentech Keynote lectures bring some of the brightest minds in the world to the ARVO annual meeting to help us think more broadly about the world around us, which in turn inspires in our work. It is a pleasure to have Dr. Thomas Cech as our keynote speaker this morning. Dr. Cech is the Distinguished Professor of Chemistry and Biochemistry at the University of Colorado Boulder and an investigator at the Howard Hughes Medical Institute. In 1982, Dr. Cech and his research group published their finding about a RNA molecule from the pond animal Tetrahymena could catalyze biochemical reactions, the first exception to the long-held belief that only protein could act as enzymes. This work led to the 1989 Nobel Prize in Chemistry, shared with Sidney Almond of Yale University. In his talk this morning, Dr. Cech will take us through the evolution of RNA research and their multifaceted function as depicted in his book, The Catalyst, RNA and the Quest to Unlock Life's Deepest Secrets. He will then discuss the potential of nucleic acid therapeutics to treat eye diseases, including Leber congenital amaurosis, as well as age-related macular degeneration. His passion for communicating science to the general public let him write The Catalyst, which The Economist heralded as one of the best books of 2024. Dr. Cech's contribution to science have been recognized by many awards, which include the Harlequin Prize of the Royal Netherlands Academy of Sciences, the Canadian and Guiana International Award, the Albert Lasker Basic Medical Research Award, and the National Medal of Science. He was elected to the U.S. National Academy of Sciences in 1987 and to the National Institute of Medicine in 2000. So please join me in welcoming this morning's keynote speaker, Dr. Thomas Cech, to the stage.
Thank you, Sophie. Good morning to all of you, and welcome to what has become my home state of Colorado. Now, today, I'm going to talk about the intersection of my area of research, which is RNA, with what all of you are interested in, which is vision and eye disease. And I had to do a lot of reading so I hope I got it right. We shall see. You can tell me afterwards. Some of you may, still be tired from traveling. Maybe you'll fall asleep during my talk. So I'm going to give you the punchline first off, okay? The punchline is that the 21st century is the age of RNA. DNA is still there. It's important. It's a little boring. It just stores information. As I will try to illuminate for you, RNA is really where all of the action in biology is. And so when I try to talk about RNA to the general public, I do start out, though, with its parent molecule, DNA, because people are generally comfortable with DNA. They know that it's responsible for your inherited traits as well as genetic diseases, that you can trace your family history through the DNA, and also that you can solve crimes with it. But when it comes to its closely related daughter molecule, RNA, some people say, well, that's much too complicated. But it really has the same information content as one of the two strands of the DNA, right? The DNA is A, G, C, and T. The RNA is A, G, C, and U, where U is informationally equivalent to the T in DNA. So it's really not that complicated. And one thing that's made it easier for me to talk to the general public about nucleic acids is that by now most people have at least heard about messenger RNA. They know that wrapped up in a tiny grease ball called a lipid nanoparticle and shot into your arm, that messenger RNA encoding the SARS-CoV-2 spike protein will give your immune system a heads up and it'll be then on the lookout for that protein. So instead of putting the protein in the vaccine, you can put the mRNA, and our bodies then do the heavy lifting of using that mRNA information to produce the protein, in this case, the viral spike protein. Nonetheless, many people have been very nervous about the mRNA vaccines. They talk about it as, many articles in social media talk about it as a medicine, as a drug, which makes people somewhat nervous. And I think instead of starting the conversation that way, we should say that mRNA is natural. It's in every cell, not only in our body, but in every living being on the planet. It's in all the food that we eat, whether you're a meat lover or a vegetarian. In Boulder, we might even say that it's organic. So, not only that, but we know how it works. We've known for 60 years that the code in the DNA is copied into the same information in the messenger RNA, and then groups of 3 of those building blocks, the A, G, C, and U, are used to specify a particular amino acid that's laid down into a chain of protein. And of course, proteins are the major movers and shakers in our body. They're responsible for our muscle movement. They're responsible for the enzymes in your stomach that are digesting your breakfast. They're responsible for our heart to beat and our neurons to work. And so, mRNA is well known. It's not something to be frightened about. We know in great detail how it works. Now, it's too early to tell if these mRNA vaccines that got such a head start during the pandemic will catalyze a paradigm shift in medicine, or maybe will have more limited applicability. Hopefully, and this is right around the corner, mRNA will provide more effective flu vaccines because they can be produced so quickly that we can figure out which strain of the flu is coming from Asia to the United States, and then make a vaccine that is specified to that strain of flu. Right now, scientists have to guess. Their guessing is not that great, and typically the flu vaccine is 30 to 60% effective per year. mRNA vaccine, we might be able to get to 95% effective. How about personalized cancer vaccines? These are in clinical trials now and are looking very promising, but we have to await the outcome and see if they're approved for human use. And then finally, a little bit higher reach would be therapeutic mRNA to replace mutated proteins. For example, in genetic diseases that could include retinitis pigmentosa and LCA. So will the mRNA be able to make enough protein to be therapeutic? Will we be able to deliver it to the right place? Well, the eye, at least, is such an enclosed compartment that I think the delivery issues are less daunting than they are for many other medical uses. So if that's all that RNA could do, to be a messenger, that's pretty cool. That might be enough to claim that the 21st century is the age of RNA. But wait a minute, I've got quite a bit of time left. RNA must do more than just serve as a message. And in fact, beyond being a message, there are dozens of categories of non-coding RNAs, RNAs that do not specify the construction of a particular protein, but instead are active movers and shakers in biology. And because of my limited time, I'm only going to be able to talk about 3 of these. One is that RNA can be a biocatalyst. We call these ribozymes, a word that we invented for ribonucleic acid that has enzyme-like activity. Secondly, RNA can bestow immortality. But don't get too excited. It's only at the cellular level, not at the level of the individual. This is the telomerase story, where RNA is an active and critical component of this chromosome and replicating enzyme. And finally, RNA can control evolution. This is the CRISPR gene editing, another RNA-powered machine where the entire specificity of CRISPR gene editing comes from its RNA component. So let's start then with RNA as a catalyst, which is the ribozyme story. So when I moved from MIT to Boulder to set up my first independent research laboratory, which I still run now, I started studying pond scum. Now, why would any self-respecting scientist want to study pond scum? Well, it wasn't any old pond scum. It was this organism, Tetrahymena thermophila, which has 10,000 copies of a particular gene. And if you're a biochemist, you like a lot of material to work with, and most of the genes in your body are present in how many copies? 2, right? One from mom and one from dad. So 10,000 is a big deal. And what we found early on was that the RNA made from this gene underwent a cutting and rearrangement process called RNA splicing in the test tube, which is very exciting because getting a reaction to work in the laboratory is the first step towards figuring out the nuts and bolts, the mechanism by which it works. But the amazing thing was we weren't adding any protein enzyme to the test tube. The splicing seemed to be intrinsic to the RNA. We didn't believe this at first. What could possibly be catalyzing it? Could it be a contaminating protein, or could the RNA really be splicing itself? And that seemed unlikely, because ever since James Sumner gave his Nobel Prize address in 1946, when he claimed that all enzymes are proteins, all enzymatic reactions discovered since then had been protein catalyzed. So this was a conundrum for a while, and this led me to this Christmas party where my graduate student, Paula, gave me this as a Christmas present. So was there a protein responsible for this reaction or not? And it turned out that we were able to prove that the RNA by itself was catalyzing this splicing reaction. Now, this excited a whole nother group of scientists that I didn't even know very much about. These are the people who were studying not Campbell's primordial soup, but the primordial soup that is thought to have existed on the planet about 4 billion years ago, when the first very primitive self-reproducing organic system arose. And when scientists thought about how this was possible, they were rather struck with a rather daunting couple of possibilities. To have some kind of a self-reproducing system, you need to have an informational molecule to hand down to the next generation. Otherwise, you wouldn't call it life. But the informational molecule that everybody's thinking about, DNA, doesn't do anything by itself. It needs protein enzymes to copy it. So now that we knew that RNA both carries information and can catalyze biochemical reactions, maybe it could be both the photocopying machine and the molecule that was being copied, the informational molecule. So maybe life started as an RNA world, a hypothesis that has gained a lot of traction in the scientific community, but is admittedly very difficult to ever be sure about, because we're talking about a historical event 4 billion years ago. So all of this led then to the 1989 Nobel Prize in Chemistry, where you can see I was a bit younger than I am now. Let's move on to the second kind of non-coding RNA, the telomerase story. So I have to start with talking about the ends of all of the human chromosomes. They are vulnerable. These are called telomeres Telos is Greek for and mir means sort of the stuff. So this is the stuff at the ends of our chromosomes. You can see it's lit up here with a nucleic acid probe. So there's a repeated sequence of DNA, which is common to all chromosome ends. And this is a vulnerable spot on the chromosome, and it needs protection, much as the plastic tips on our shoelaces protect the shoelaces from unraveling. And those plastic tips are called what? Aglets, I heard someone say. Yes, you must be a crossword puzzle aficionado. So this telomeric DNA in our somatic cells shrinks as the cells divide. And when the telomeres get very short, this is a signal for the cell to stop replicating. And that's a good thing, because once your tissue becomes large enough, once you as an organism become large enough, you don't want to keep dividing. And it's thought to be a cancer preventative mechanism to have a sort of a break on unlimited proliferation. But there are some cells in our bodies, like the stem cells, that need to continue proliferating, right? And so they have to, even in an adult, so they need a way to extend this DNA at the chromosome end. And this is the telomerase enzyme discovered by Liz Blackburn and Carol Greider, who got the Nobel Prize for that discovery. And they also discovered that there was an RNA subunit that was critical for the action of telomerase. It's shown here as the purple ribbon. And among other things, that RNA serves as a template to determine the order of nucleotides of DNA that are laid down at our chromosome ends. But the RNA is not, in this case, a catalyst by itself. A protein is also needed, and that was discovered in my lab in Boulder by a fantastic postdoctoral fellow named Joachim Lingner, who's now running his lab in Switzerland. And we called this telomerase reverse transcriptase. Many of you have heard about it. It goes by the shorthand name TERT. And it's the combination of this TERT and the RNA that powers the addition of the DNA at the ends of our chromosomes and gives cells immortality. Now, a lot of people think that's a good thing. In fact, maybe you can live longer if you lengthen your telomeres, and you can buy various food supplements on Amazon.com, for example, that are guaranteed to lengthen your telomeres. They're clinically proven. But I must say, these have not been through FDA placebo-controlled tests because they are sold as food supplements. So in fact, they have not been through clinical trials. But if they did work very well, it might not be a great thing, because what is one kind of a human cell that continues to proliferate in an uncontrolled manner? Cancer cells, right? And so they need telomerase for their own immortality. 90% of all human cancers find a way to reactivate the telomerase enzyme that is normally turned off in adult cells. And this guy, Levi Garraway, and his Harvard Medical School fellow, friend of mine, Franklin Wong, about a dozen years ago, discovered mutation in the promoter, in the regulatory region of the gene we had discovered, the TERT gene, that drives cancer in a major way. And in fact, all over the world, there are about 400,000 new cases of melanoma each year, and 80% of those have independently, through mutation, gotten this change in the promoter region of the TERT gene that allows telomerase to gain a new foothold and to power the immortality of the cancer cells. 80% of glioblastoma has that same base pair change, 70% of bladder cancer. And in the eye, a very large percentage, this is huge for an oncogene, right? About 40% of conjunctival melanoma is also driven by this same mutation in the TERT promoter. It's rare in uveal melanoma, but other mechanisms are there to help activate telomerase, epigenetic mechanisms, rather than this promoter mutation. So we are working then to try to find inhibitors of telomerase that could be therapeutic for cancer. And finally, let's go on to the third kind of non-coding RNA, one that can do genome editing, and this is the CRISPR system. Now, genome editing, when I talk to non-scientists, and I apologize for being too simple for most of you here, but I think it's useful to use sort of an analogy. So genome editing is like text editing, but in a normal font size, all of the DNA in each of a human cell would take about 1 million pages. I actually calculated this. 1 million pages to have your whole genome as a Word document. And so genome editing means to have some kind of software that will search through the 1 million pages and find a typographical error. In this case, highlighted in red, the letter mux should be makes, and after editing, then the correct sentence is restored, right? And so this was something that was discovered by Emmanuel Charpentier and Jennifer Doudna. They got the Nobel Prize in 2020 for their work on CRISPR genome editing, came from bacteria, but can be repurposed to work in human cells to do the kind of text editing that I just talked about. We were very excited in Boulder, particularly when Jennifer won the Nobel Prize, because she had been a postdoctoral fellow in my laboratory a few decades ago. Here we are at a lab reunion a few years ago, and the people next to her are also faculty at University of Washington, University of Chicago, and at the Scripps Institute in La Jolla. Now, how does this CRISPR work? Well, it's not quite as easy as this red scissors cutting the DNA, but it does cut the DNA, and that seems dangerous, but it cuts it exquisitely precisely because it carries around a guide RNA with this molecular machine, and it's only when 20 units on the guide RNA match and compare with one strand of the DNA that it locks down, and then a cutting activity, which is in an associated protein, in this case the Cas9 enzyme, cuts both strands of the DNA. Now, why would that be useful in research or therapeutically, to even if it's highly specific, to cut DNA? And that's because damaged DNA is very deleterious for biology, right? And so even from the most primitive organisms, there's always machinery in every cell that repairs cut DNA. And it's during this repair process that new sequences can be inserted into a chromosome. And for example, if there's a genetic mutation, this can be used to reverse that and return it to the natural or the wild type sequence of the DNA. Now, this kind of genome editing is already an outstanding research tool. We've been using it since 2012 in my laboratory. Even undergraduates have performed it quite successfully. But how about the medical potential? Well, deleting bad genes from the genome by cutting on both sides and letting the DNA be repaired is occasionally useful. You might imagine that vascular endothelial growth factor, that is a problem in wet macular degeneration, would be a place where you might want to remove that VEGF gene to prevent the blood vessels from going across and obscuring the retina. But maybe more potential would be to repair faulty genes, because usually we don't want to get rid of a gene, we want to fix a mutation in it. This, we think, has enormous potential because most diseases are either genetic or have a genetic component. And often, now that we have the human genome and very rapid and cheap DNA sequencing, genetic counselors can find the cause of a human disease, but they haven't been able to fix it. Now with CRISPR, we might be able to provide a platform where we can correct mutations in the human chromosome. So in summary then, I've given sort of very crash course here in RNA biology, right? RNA can be a message which is critical for all of life, but also can be used therapeutically for vaccine development. But beyond being a message, RNA can serve many non-coding functions. For example, it can be a ribozyme, it can be telomerase, part of telomerase to bestow immortality at the cellular level. It can control evolution through CRISPR genome editing, another RNA-powered activity. But now let's turn to something that is of more direct interest to this audience, and that is the possibility that RNA therapeutics could have an impact on eye disease. And I'm going to talk about 3 different kinds of therapy that we might want to achieve. One would be to knock down or to inhibit a disease-causing protein. The typical nucleic acid therapeutics that are used here would be either ASOs, which is an abbreviation for antisense oligonucleotide. Antisense not meaning that it doesn't make any sense, but meaning that it can pair with a messenger RNA and affect the function of that messenger RNA. There are also small interfering RNAs, which tap into a system that is intrinsic to already in all of our cells called RNA interference, but the siRNA can direct this natural activity, again, to inhibit the production or the amount of a particular mRNA in a cell in the eye. Secondly, you might be able to want to restore a mutated protein. This can be done in principle with CRISPR gene therapy to repair the mutated gene. Now, antisense oligos can also be used to restore a mutated protein. Wait a minute, I just said they would inhibit it, they would knock it down. How can they do both? Well, we know so much about RNA biochemistry that we know that there are some places where if there are a lot of mutations in a particular part of a messenger RNA, we can add an antisense oligonucleotide that will cause the splicing machinery to jump over that mutated part. And then the protein is missing a chunk of its amino acid sequence, but through studies you can decide whether that protein is still functional, and often it still is. And so this is a way then to not have the mutation in the final protein product of the messenger RNA. And finally, we might be able to restore the mutated protein simply by adding the messenger RNA for the correct protein and thereby compensate for the fact that the protein is mutated. And the third category of RNA activities that have been used in the eye is to use RNA to block a binding interaction, an interaction where 2 macromolecules are talking to each other. And this is sort of like an antibody, but it's made out of RNA. We call these aptamers. And although they're not widely used now, they might be in the future, I want to highlight this because it has a Boulder, Colorado connection. The very first RNA aptamer ever used to successfully treat a human disease was developed in Boulder. It's called pegaptinib, and it was developed by a friend of mine, Larry Gold, in his company. And it was used to block the blood vessel growth across the retina to target wet macular degeneration, and it was successful. At the time, it met an unmet medical need. It was approved by the FDA for use in 2004. And it had a very good binding constant. Its equilibrium binding constant was in the picomolar range, so it bound very tightly to the VEGF. It also had a pretty good lifetime in plasma, 18 hours after polyethylene glycol was added to its end. You can see this, the structure of it here. It's just a small piece of folded RNA. And so it was approved for medical use, and did help a lot of patients with wet AMD, but then it was displaced by more effective monoclonal antibody anti-VEGF treatments shortly thereafter. Nonetheless, it proved the principle that aptamers could be of therapeutic value in the eye. So what's up next in terms of RNA therapy for AMD? Well, I think siRNAs against VEGF are already in preclinical development, and certainly CRISPR knockout of the VEGF gene in the retinal epithelium is another approach that is being really pursued currently. So before I go on, let me just take a minute to compare this gene therapy with RNA therapy, because you've heard a lot about gene therapy, that's DNA, right? Why DNA or RNA? Well, gene therapy, whether it's delivered by adeno-associated virus, AAV, or CRISPR genome editing, is a permanent fix. One shot in the eye, and that might be all the treatment the patient would ever need. So that is seems to be better than an RNA therapy, which would have to be re-administered perhaps every few months if it's an mRNA, an antisense oligo, or an siRNA. On the other hand, we aren't yet sure that these gene therapies are 100% accurate. And if you do CRISPR genome editing and you repair the wrong part of a chromosome, a so-called off-target effect, that can be very deleterious. And so the RNA therapy has the advantage that it's not a permanent change in our genetic makeup. It's reversible, just like a lot of medicines that we take. If the patient isn't doing well on it, we quit giving it to the patient, right? So I think for the moment, it's best to pursue both the gene therapy and the RNA therapy to see which one will be the most safe and effective before we throw one of these away and work on only one of these approaches. Now, in terms of the gene therapy, I'm going to compare gene therapy in the eye with the one CRISPR genome editing gene therapy that is already approved for medical use. And this is against sickle cell disease, a terribly debilitating condition that is really the scourge of particularly the African American population. And we've known for many decades, it was the first human gene where the precise cause of a genetic disease was actually determined. And it's a single base pair mutation in the beta globin gene that makes one of the components of hemoglobin. And that single base pair makes a glutamic acid in the normal globin protein, instead being a valine. Well, it's only one amino acid difference, so it works pretty well under most conditions, but when a person gets stressed or low oxygen or other kinds of perhaps personal stress, then this one amino acid change makes the hemoglobin clump together, and it distorts the red blood cell into this sickle shape shown here, and that sticks in the capillaries and is excruciatingly painful and prevents people with this condition from holding down a normal job or raising their families in some cases. So the CRISPR genome editing is perfect for this because we know the mutation, and every patient has that same mutation, and so one therapy will suit everyone. Now, with Leber congenital amaurosis and other inherited eye diseases, it's much more difficult. Why is that? Because in these diseases of inherited retinal photoreceptor dysfunction, we have multiple disease genes. And so we need genetic testing, and we have to have precision medicine for each therapy to be just for that subset of patients that has that particular type of LCA. So in the case of RPE65 LCA, there's Luxturna, which is an adenovirus-delivered gene therapy, is already approved for use. Probably some of you are using this and know a lot more about it than I do. CEP290 LCA, there are clinical trials already ongoing to use CRISPR-Cas9 genome editing to correct the mutation in this disease. There are also antisense oligonucleotide therapies. Again, this would be a positive use of antisense. It binds to the pre-messenger RNA and corrects a faulty RNA splicing, which is caused by the mutation, to now correctly splice, give you a correct messenger RNA, which then gives a correct protein. And in the case of GUCY2D LCA, adeno-associated virus gene therapy is again in clinical trials. Now let's move on to retinitis pigmentosa. It's an inherited progressive retinal degeneration, as you all know, but there again are multiple genetic causes, something like 100 genes that can cause RP. And so genetic testing, again, is required. There are RNA therapies, antisense oligonucleotides, that can be used to either destroy an aberrant messenger RNA in the case of the Rho mRNA and therefore prevent the protein from being made, or to skip over a mutant exon in the case of Usher syndrome 2A and restore a positive protein. Gene therapy is approved in this case to restore RPE65. Pretty good for those individuals who have that particular RP, but it's only 0.3 to 1% of the retinitis pigmentosa. So that's sad, right? That means we have to have a lot of versions of this in order to really make a general impact on those suffering from going blind. And mutations in non-coding RNA. So you think about proteins as being the problem in disease, but how about these non-coding RNAs that I've been talking about? So I was very excited to see very recently a paper coming out of Suzanne Roosing's laboratory in the Netherlands, published just a few months ago in Nature Genetics, that she has found, and her team has found, mutations in a non-coding RNA that cause retinitis pigmentosa. In fact, there are two different non-coding RNAs, ones that I'm very familiar with because they're involved in the machinery that does the RNA splicing. So these are the U6 or the U4 small nuclear RNA, and the location of the mutations is around this bulge in a place where these two non-coding RNAs pair with each other. During the RNA splicing cycle, they need to both pair and to unpair at a particular time, and these mutations make the pairing too tight, so they can't effectively come apart to do their jobs. And so it could be that RNA therapy against this RNA target, for example, ASOs to destabilize and tweak the stability of this mutant pairing, or perhaps we could deliver excess normal snRNA as a therapeutic to these patients. So this, again, just shows how broad we have to extend our thinking to really make an impact on a lot of these eye diseases. I just in closing need to say a word about AI, because it's the big truck that's not only coming down the highway at us, but it's already here, right? And it's already transforming both research at the University of Colorado and at all other research institutions in the country. Our students are using it in diverse ways, but also it's transforming medical diagnosis. It is really good at image analysis, which is something you all care a lot about, right? And you can all look at this fundus from diabetic retinopathy and say, oh yeah, that's easy. But what if it were a much more subtle lesion? Then AI can very accurately determine these, and the human, in trials, it's actually sometimes outperformed ophthalmologists in terms of diagnosis. So I think this is something that we should all embrace and get to know about and use, but also to be skeptical about. Because the advice that we have a new AI and genetics PhD program that I've started at Boulder, and the advice I'm giving the students is, yes, learn to use it, but always be skeptical and validate the answers that AI gives you independently, because hallucinations are very common with AI. And so, in closing, I hope I've given you some reason to believe that maybe the 21st century is the age of RNA. This is something that I tried to bring to the non-scientific general public with this book, The Catalyst, published a couple of years ago by W.W. Norton. And am I making any progress in getting the general public interested in RNA? Well, maybe a little bit. I don't know. My wife showed me this a couple of months ago, earlier this year, crossword puzzle from the New York Times, and 28 across is molecule that can self-splice. And so it's 3 letters. What must those 3 letters be? Please. N-A. Yep, you got it. So with that, I want to thank my research groups through the years that have done all the work that I enjoyed talking to you about tonight, and also thank you for your attention. And I'd be glad to answer questions.
That is amazing lecture, so inspiring. It's a perfect example of how to challenge assumptions and lead to discovery that transforming our life and our science. So before we go on the Q&A, I have a little surprise for our audience today. When you enter the room, some of you received stuffed bull bears as a gift. Some of the bears are holding little books. If you have a bear, check it now. Check it now. If a bear with the book, you will receive a complimentary copy of Dr. Cech's book, The Catalyst. In addition, each copy has a signed book plate. However, Dr. Cech is kind enough to be available to sign the personal messages in middle after this session. Just outside the session door, you can get in line to get a signed personal message and to meet Dr. Cech. Now we do have a few minutes. I welcome the floor to come to the mic to ask any questions. There are 4 mics, 2 over here and 2 on the other side. And I cannot see. Okay, I think we see someone on mic number 4. Could you identify yourself first?
Hi, Jack Sullivan, University of Buffalo. Dr. Cech, it's wonderful to see your presentation. I I had a question about catalytic RNAs, right? So antisense and RNAi use protein machinery to cut the target, right? So if we look back in time to, say, Ribozyme Pharmaceuticals, Inc., right, which were using minimal hammerhead ribozymes extensively modified with systemic injection, their clinical trials didn't move forward. But with the new structural biology about extended hammerhead ribozymes, it seems that the better magnesium performance and rates could lead to a revitalization of true catalytic RNA therapeutics. I wondered about your perspective for that.
Yes, well, your level of scientific knowledge about the topic is right on target. Certainly, I think when we started this RNA company in Boulder many decades ago, we shrunk down the size of the RNA below the size where it really was optimally active in human tissue. And so if we had it to do all over again, we would make it, as you already said, larger so that it would be more active under intracellular conditions. And there are a few companies that are still pursuing ribozyme technology. I think that to a large part, the only thing that is left over positive from my experience with this company is that we developed a lot of ways to stabilize RNA, to chemically modify it, to make it more useful as a therapeutic. And that technology is still being used, for example, by Alnylam, the siRNA company in Boston. But in terms of RNA therapeutics, a few companies are still trying to do the ribozyme, but it's been largely left in the shadows as newer entities have come forth. So thank you for your question.
Thank you.
Great. The next one will be number 4 again, and then the next will be number 1.
Hi, Tom. My name is Matias Alvarez, Chief Scientific Officer of Nano Neurosciences in Florida. I agree with your comment that messenger RNA, ASO, and siRNAs are very efficient and much more safer than gene therapy. However, one of the challenges that remain are delivery vehicles, and I wanted to hear your input regarding that. I come from the world of lipophilic peptides that self-assemble into nanostructures, and thus I propose that these are very efficient vehicles for RNA delivery.
Yeah, absolutely. You've put your finger right on it. I think that delivery is the big challenge for these RNA therapeutics. And that's where, for eye disease, there's a real advantage, is because it's such a closed, defined compartment, and very different from having to inject an RNA therapeutic into the bloodstream and hope that it traffics to the right tissue. They usually go to the liver, as you know, which is fine if you want to treat liver disease, but not good if you want to treat a muscle disease. So I think that appending various entities to the RNA, your technology sounds very promising. There are many others that are being tried right now to make the RNA packaged up in a way that is still going to be active, but will find its right place to do its job. That's the big challenge. And thank you for being one of the people who's working on this.
Thank you.
Thank you. Mike, number 1.
Okay. I have 2 questions. This is Ping Wu Zhang from Johns Hopkins. The first question is, when double-strand break in a cell, how the cell responds to that? The insertion or deletion is a random or it's a text determined? We found that sometimes it's not random. This is question 1.
Yes, well, again, to talk about all of the DNA damage repair pathways would be more than an hour of a talk in itself. But there are two major kinds. There's a sort of a quick and sloppy kind of repair, which is just stitching the broken chromosome back together. And this is called non-homologous end joining, or NHEJ. It tends to leave mutations at the site of repair. And that's one thing we have to worry about with the CRISPR genome editing, is that if it doesn't insert the correct DNA in the damaged spot, there's usually a scar that is left, which is hard to find, but we can find it with DNA sequencing and say, uh-oh, we've actually damaged the DNA at the site of cleavage. Now, the activity that we want is called non-homologous end joining. It's where a donor DNA template will be substitute in for the missing part of the DNA at the cut site and do a repair at that site. And that scientists are working hard to drive that accurate repair instead of this quick and dirty non-homologous end joining. So there are many companies that are working hard on this, and I think the technology is improving, but the safety is getting really high, but I think, to be fair, it's not 100% safe yet.
Okay, the second question, you mentioned the importance of non-coding RNA, and more evidence indicates non-coding RNA actually some of their coding RNA, specific RNA, right? So should we redefine the definition of non-coding RNA in the future?
Yes, you're absolutely right that some of the RNAs that were initially classified as non-coding turn out to have short open reading frames to make peptides, which could be sort of have hormonal kind of activity in cells. I think those should now be called messenger RNAs, and they shouldn't be called non-coding RNAs. But it does get complicated because there are some RNAs that may have both a non-coding RNA function and code for a small peptide. One activity in the cell nucleus, the coding activity in the cytoplasm of the cell where the protein synthesis takes place. So maybe we do need some new nomenclature. Thank you for that.
Great. Another questions from number one again.
Hi, that was a really great talk. I'm over at Seattle Children's Research Institute. I wanted to ask your opinion about different types of gene augmentation using RNA technologies, specifically with self-amplifying RNAs and circular RNAs.
Yes. Well, the circular RNAs, the reason for making these is because they don't have ends, that's the definition of a circle, and a lot of the degradation of RNA takes place from an end. So circular RNAs are much more stable, but still quite active in biological systems. And so that is a very useful direction for the field. Now, the self-amplifying RNAs, this again for vaccines could be a way of getting additional copies, a big augmentation of the copy number after an initial injection of, for example, a vaccine. And is this safe? I mean, could the self-amplifying actually amplify other RNAs, and might that be deleterious? All of these potential therapeutics have to pass very rigorous safety studies, right? Sometimes we can't predict whether something will be completely safe or efficacious, and that's why we have, or perhaps used to have, an FDA that would help us make these decisions.
Thank you.
Yes, FDA is important. I saw initially a mic too. Someone wanted to ask a question. If not, I want to ask the last question. We had 2 more minutes. Is that in terms of the gene or CRISPR therapy, does the eye present like special challenges or opportunities? What do you think about the field, direction of the development of the field?
I think more special opportunities. And in fact, when I was president of the Howard Hughes Medical Institute, we were bringing new investigators into HHMI to support their research. And the first gene therapy person we supported was at CHOP, the Children's Hospital in Philly, and she was working on gene therapy in the eye. And she argued very persuasively that this is a way that you could actually be pretty sure that your therapeutic entity was staying where you injected it, right? So I would say much more opportunity, but challenges are always there as well. Thank you, Sophie.
Yes, definitely, because each indication might require patient-specific. That's why precision ophthalmology is so important, is patient-specific. Great, thank you. Is no more question? Then thank you so much for your talk. So.